So we're here at the iwNHL, and we just finished a session on novel combinations in lymphomas. I'm Laurie Sehn from BC Cancer, and I'm joined here by my colleagues, Dr. Juan Alderuccio from University of Miami and Dr. Gilles Salles from Memorial Sloan Kettering. And I have to say that just my general impression of the session is that we are being overwhelmed with new data that really is, I think, changing how lymphoma is being managed in real time. And we spent a big part of the discussion reviewing some of the novel agents that have now emerged as standard of care, and some of the exciting trials that have moved forward with these novel agents that will have data forthcoming. I in this session discussed the new data of Tafasitamab combinations, where we know it was initially approved in the InMIND trial for relapsed refractory DLBCL and is now available out as a platform for patients in the relapsed refractory setting. But more recently, we had the InMIND trial, where it was compared in a head-to-head trial against R-squared for relapsed refractory follicular lymphoma, which has recently earned it an approval. So the combination of Tafasitamab, lenalidomide, and rituximab now is a new approved option for patients with relapsed refractory follicular lymphoma after at least one prior line of therapy, because the head-to-head trial demonstrated an improvement in response rates, CR rates, as well as its primary endpoint of progression-free survival. And the comparable toxicities were relatively similar, maybe with a slight increase in viral infection that were generally mild, but is now available as an additional option. And then we heard other discussions. Juan, you discussed some of the really exciting data on Loncastuximab.
Yeah, thank you, Laurie. We discussed the Laurie Sehn clinical trial, which combines Loncastuximab with glofitamab in patients in second-line plus large B-cell lymphoma, showing in the initial analysis of 40 patients, no new safety signals with this combination. And in a preliminary analysis of 30 patients, a high response rate that at the time of data cutoff appears to be durable. Now this study is expanding to enroll a total of 100 patients, and we expect to have this recruitment finalized by early 2026. And we also discussed data on follicular lymphoma with Loncastuximab in combination with rituximab. This was initiated as a single-center study, and response also were high in the relapsed refractory setting across different high-risk groups. And now the study transitioned to a multi-center study, also aiming to enroll a total of 100 patients. And we, in an updated analysis with a data cutoff in mid-September, we observed high responses and durable responses with a 2 years PFS about 80%. So we also hope to finalize the enrollment in first quarter of 2026 and report this data at ASH next year.
So that is a really, I think, exciting combination for follicular lymphoma, and maybe a little bit surprising, but it's nice to see that we're going to have even more data on additional patients moving forward.
Yeah, and also what we are doing in this multi-center phase to fix the treatment duration to 6 cycles, aiming to decrease the toxicity associated with cumulative doses of Loncastuximab.
Mm-hmm. Good. We certainly look forward to that. And Gilles, you reviewed some of the data on Polatuzumab combinations, and I have to say the discussion was quite lively.
Yeah, I mean, Polatuzumab vedotin, as we know, as drugs that has been combined with bendamustine rituximab. You did the study with RCHP, withdrawing vincristine, and now is being developed in combination with bispecifics. I think there are two parts of this discussion. The first one is the differential effects that was observed in the POLARIX study, where patients with a genetically determined ABC cell of origin appears to have a very large benefit of this drug, with almost 30% difference in progression-free survival and more than 10% in overall survival, while there was little or no apparent benefit in the other subgroup. And how do we translate that? How do we apply that in clinic? Was it detrimental to some patients? It's a little bit unclear. There are also probably some other patients with GCB phenotypes that may benefit and others not. So there is an open question here, and we heard during the meeting in the spring that in the Polago study that used Polatuzumab vedotin in combination with our GEMOX, there was no difference in and showing an overall survival and progression-free survival benefit. There was no difference in patients that were GCB and ABC. So there is still complicated data here, and I think people may choose one way depending on what is labeled in their country, depending on what's authorized, and things like that. But I think it raised, to me, more fundamentally the question of how do these drugs work? What explains the efficacy? Is that the binding to the antigen? Is that the internalization? Is that the activity of MMAE? And the data experimental for Polatuzumab suggests it's the antigen expression. But in the same session, we heard this trial of brentuximab vedotin plus R² in relapsed refractory DLBCL, showing that in patients with virtually no CD30 expression, there was a benefit of adding this drug. So that's one of the questions, more efficacy and the second one is, we have all these trials being developed in the frontline study. We have the efficacy in the second line of bispecific plus Polatuzumab vedotin is the most soon in a randomized trial, or Glofit in phase 2 trial. And we have, as just said by Juan, the Loncastuximab plus Glofit. So how are we going to sequence that? And it will depend, probably at this time we have R-CHOP and Polar-CHP approved in the US and available in other jurisdictions with some limitations. There are a wave of about 6, 7, 8 randomized trials that are going to be delivering results with Tafasitamab, with acalabrutinib, with bispecific antibody in the next 1 to 2 years. And how are we going to choose? How are we going to optimize what we give to patients in terms of toxicity, durability? And I think that's an interesting question, and also the question of sequence. If you use bispecific before, what are you going to use after? If you lose MMAE-containing antibody-drug conjugate, such as, for instance, Polatuzumab, can we use Zilvo, which is a WOR1 MMAE-containing drug in the relapse setting, which has also interesting result, or should we use a different toxins and that one? So I think all these questions are now emerging. Some have data to answer, some are data to be confirmed, and I think we'll need to understand. It raised a question that, what is best for our patient beyond what is approved and how to best sequence, and that will probably keep us in a very lively situation in how to optimize the treatment of patients with B-cell lymphoma.
For a very long time. One of the things we also heard in this meeting was a very nice review of DLBCL genetic classification is delivered by Margaret Shipp. And, I think there's such a paucity of data in terms of using it as a predictive classifier in the relapse refractory setting. I mean, many of our relapse refractory trials have maybe been a little too small to probably, sort of mine that kind of predictive data, but we now have some larger phase 3 trials that have just emerged, and I think we still have yet to see any of this potential correlative analysis that might make us a little smarter in terms of which patients are preferentially benefiting in the relapse refractory setting. But I think that's going to be essential to be built into all trials so that we can at least, go beyond, our very rudimentary practice of sort of empiric management.
I will emphasize that, Dr. Margaret Shipp gave this beautiful presentation from the paper that was recently published and what was done in Polarix and other questions here, and how to implement this genetic classification in clinics remains to be determined. But I think it's probably relatively urgent, because if we assume, and maybe we are wrong, but that we may have an MMAE-containing agent in the first line with chemotherapy plus a bispecific, the patient that will unfortunately experience relapse will have maybe some quite specific particularities, and we may have used CD20-directed therapies, MMAEs, others, And we may have to go back to something we have a little bit neglected in the last 5-10 years, which are targeted molecules, given the Phoenix and Robust data. But we know that we're making progress with these targeted molecules. Maybe again, there are combinations that are here. So I think continuing to explore the genetic of this lymphoma is important, because we may identify some subset that may benefit particularly of some of these drugs.
Juan, maybe I'll ask you a question. So most of us, are looking to get away from maybe further chemotherapy, at least in the relapsed refractory setting, with these novel agents that are now available. Yet we've had this series of clinical trials that have really been built on a GEMOX backbone. Yeah. In your clinical practice, are you eager to give the GEMOX platform with these novel agents? Would you, if you had access to GlowFit GEMOX, would you give it, or would you be just as happy to maybe jump right to GlowFit alone?
So yeah, in the United States, GlowFit GEMOX is in NCCN guidelines, so we have access. And that has been my practice in those patients that for some reason are not eligible for a clinical trial, and use as a standard of care the combination of GEMOX with glofitamab, and helped with few patients and with excellent responses and very quickly. So it's unfortunate it has not been approved, and hopefully the sponsor can provide the necessary data to have the regulatory approval. But definitely, I think it's a very potent combination that will improve patient outcomes.
If you look at the bispecific antibody glofitamab and epcoritamab, which had exactly the same CR rate in their pivotal phase 2, 39-40%, you see that when combined with either cytotoxic agents, such as GEMOX with glofitamab, but also with epcoritamab, the epcoritamab GEMOX phase 2 was published, or with an antibody-drug conjugate, Lonca, Pola, and maybe others in the future, you raise the bar of complete response by 10-15%. It's a little bit to be determined if this is the same CR that lasts. I was encouraged by the update of the STARGLOW, the trials that compare glofitamab GEMOX versus our GEMOX, showing that it looks like after 1 year, when we were off therapy, the curve between 1 and 2 years didn't change much for those patients in CR, suggesting that maybe some patients in the relapse setting may be cured by this regimen. Because that's the main question when we have CAR T available, is that are we offering bispecific combination, or are we sticking to CAR T because we have 5 years data with CAR T? Again, interesting question for future debates.
So maybe we'll just finish up by, I'll ask you, of the agents that we discussed in this last session, which were largely antibody-based therapies, what trials that are ongoing are you most excited about seeing the results from? Gilles?
Well, I think that the trials with currently Polatuzumab vedotin, which is a drug I discussed, are available. Polarix was just published with 5 years update. We like to see the publication of the Mosun Pola, which probably is quite practical in the community, and the rest are phase 2. The Polago trial, it's a combination in relapse setting. I don't see really where it fits in the armamentarium. So I would like to see a little bit more data with the Mosun Pola, and I will see what other combination will be developed and available as comparative trials.
Yeah, I think also I agree, and also the frontline study with Polarchip plus glofitamab versus Polarchip, that's going to be very interesting, and see, because we're going to build most likely future trials based on that data. And also the combination of Loncastuximab, glofitamab, I think is interesting. After we expand the current cohort to 100 patients, and we have longer follow-up data, and to see how durable are these responses. And I think also the ctDNA data is going to help until you have longer follow-up to see how deep are the responses, and if we can start to challenge some standard of care like CAR T-cell in the second-line setting.
Yeah. Well, I think we're going to have no shortage of, things and controversies to discuss in the future. I think the next big trial to read out at least the big phase 3 trial will likely be the frontline trial, which you pointed out, which will compare Tafasitamab, lenalidomide, and R-CHOP versus R-CHOP. So to be continued, we'll see. I think it's really a quickly evolving area, but the exciting part has been that, you know, all of these options that have opened up and are likely to open up are really showing an impact in terms of favorable patient outcomes. Well, thank you for joining me. You know, a very interesting discussion.
Thank you.
Thank you for having us.